Welcome everyone to day 3 of the Brain Cell Atlas from Data to Knowledge workshop. I'm David Panchison from the National Institute of Mental Health. We've had some exciting sessions the last 2 days. They were focused on the approaches and challenges of generating a brain atlas and an ecosystem for organizing that atlas data into coherent knowledge across dimensions and scales. Moving forward, today's first session is focused on how brain cell atlas knowledge can be utilized for understanding brain biology and health and disease. I'm happy to introduce our speakers for 2 keynote presentations in the morning sessions, and these will provide examples of utilizing single-cell based data for exploring molecular and cellular pathways of pathophysiology. And time permitting, we'll have a Q&A after each keynote presentation, so please raise your virtual hand. The icon is on your MS Teams link if you want to have your microphone activated. Alternatively, you can use the chat function to have your questions answered via chat. So first, we'll have a joint presentation by Dr. Steve McCarroll and Evan Macosko. Steve McCarroll and his lab are working to understand how human genes and alleles give rise to brain disorders such as schizophrenia, bipolar disorder, and Huntington's disease. They discovered the contribution of the complement genes in schizophrenia, the way that somatic mutation leads to the onset of Huntington's disease, and when Evan Macosko was in the lab to conceive it, developed a droplet-based single-cell RNA-seq method known as DROP-seq. Evan Macosko and his lab developed and applied genomic technologies to study brain diseases by measuring gene expression, quantifying synaptic connections, and detecting molecular interactions. He began as a postdoctoral fellow with the development of DROP-seq, and then his group has developed slide-seq and slide-tag methods to enable genomic measurements at high spatial resolution to characterize cellular responses and identify disease-vulnerable cells in neurodegenerative diseases. So Steve and Evan, welcome. The floor is yours.
Yeah, so Steve and I are just very pleased to be able to present for you an update on our center's ongoing work to characterize how cells of the human brain vary across regions and people. Our project really kind of recognizes and tries to embrace the transition that we are undergoing with this phase of the Brain Initiative from generating atlases of highly homogeneous laboratory animals to humans. And humans intrinsically, can't be rendered uniform as we have been able to do with laboratory animals. We can't inbreed them, and we can't grow them in controlled environments. And so we know by nature that the cells that we're surveying from individual, cells from individuals can't have a fixed gene expression, and that gene expression has to vary both across, environmental perturbations, but also across genetic variation. So our team is really trying to build a next generation atlas resource that can kind of leverage these additional sources of variation to inform many different areas of human neurobiology, including neurogenetics, translational neuroscience, and also systems and circuit biology. So our resource really has 2 parts. The first is that we want to learn the consistent and conserved cytoarchitecture of the brain. And we're doing this using single nucleus resolution spatial profiling using our slide tags technology that I'll talk about in a minute, that we're applying to dozens of donors in key brain areas that we know are very important for disease. I've given some examples here. And that really, we hope, will teach us which cellular and molecular features of these tissues are conserved across people to provide a kind of consistency map that we can use to then inform the second phase of our project, which is to leverage the higher scale of pooled single nucleus RNA-seq experiments to reach hundreds of donors across 50 brain areas representative of the brain as a whole to understand principles of variation, and again, using this slide tags data as an anchor. We've assembled a really amazing team to do this, and I just wanted to take a minute to recognize them. We don't have enough time to go into all of their contributions, but led by myself and Steve and also Kiku Ichihara, we've assembled a team of neuroscientists, genomics technologists, chemists, and computational biologists to generate a rigorous, consistent spatial and single-cell genomics data set across a very large number of donors. And we're just really grateful to have such a motivated and hardworking team that are really passionate about what this resource can deliver. So today, we'll focus our team's findings within 2 domains. these kind of consistent domain of slide tags data generation, and then Steve will talk about the single nucleus RNA-seq work that really helps us to understand the effects of common genetic variation on gene expression within the striatum. So I'll start with telling you about the slide tags work. And just to give a brief introduction to the tissue in which this work has been done is the human striatum. And there's a lot to say about the striatum, but it's a hub for processing many different neural streams spanning motor, cognitive, and limbic systems. It's a really frequent site of neurodegeneration and of vulnerability to psychiatric disease. And prior work has identified molecular, physiological, and disease-vulnerable gradients across the striatum, but a cell-level cytoarchitectural map has not really been generated. And so one particular challenge has been that single-cell and spatial tools are deployed on a small number of donors, and it makes it challenging to know which of the spatial features one observes are highly conserved and which ones might be more specific to individuals' neurodevelopment or experience. And so our goal for this first project that I'll talk about was to leverage our spatial genomics platform performed on multiple postmortem brains, spanning both sexes and across lifespan, to define striatal architectural features that are conserved and reproducible across individuals. So we applied our slide tags technology to the anterior striatum initially to do this across 19 donors and across the adult lifespan. And crucially, the striatum is really large, and our slide tags approach really allows us to get single nucleus RNA-seq quality data with high spatial resolution across this full size of the striatum. In total, in this data set that I'll talk about, we gathered 1 million striatal profiles from 19 donors, and each of these donors covers 7 square centimeters of striatal area per donor. So I'll be telling you about analyses that were led by Matthew Lee and Andrew Kraft to characterize the cellular architectural features that we see in these 19 donors. And so we began by utilizing the Allen Institute's new basal ganglia taxonomy. So we divided the cell types into those major groups that were defined by the current HIMBA initiative. And then we ran unsupervised clustering on individual cell groups to see if we could discover whether there are certain, consistent cytoarchitectural features that we could see. And so here I'm showing in this first UMAP plot a representation of the D1 matrix MSNs. These are the primary neuron types of the striatum. They're also called SPNs. And one major distinction is in the expression of D1 dopamine receptors versus D2. So we're looking at those that express the D1 dopamine receptors. And some of these cells also are labeled as matrix cells. That's the majority, and a minority have this spatial pattern that are called patches that have a different connectivity. So we're looking just here at the D1 matrix cells. And we just did an unsupervised clustering, dividing these cells into 6 groups based on their gene expression commonalities. And when we looked at the positions of these particular populations in the striatum in each one of these donors, we found very strong consistency in where they were localized. And these patterns followed some features that we know from neuroanatomical division. So there's some that are specific to regions of the caudate, but there are some that also span multiple areas. For example, on the top here, the zone 1 has a shared representation from both the putamen and the caudate. So these are sort of novel cytoarchitectural divisions that were not appreciated in the sort of original anatomical division of the primate striatum. So you can see that it kind of follows this circle, and so the zones kind of go from the most dorsal, being the zone 1, and then as we kind of move our way around the circle, we go in a kind of counterclockwise fashion from the caudate down to the beginning of the accumbens and around to the putamen on the right side. One really interesting thing always in single cell analysis is how do we know how many clusters to generate? And how do we know if a certain number of clusters is consistent? So an analysis that Andrew did that I think is quite compelling is he just iteratively added more and more clusters to this unsupervised data, starting with this 6 zones that you see here, where you can see that the cells are quite nicely separated and they're not overlapping with respect to each other. But as you add more zones in each of the donors, you start to see that these zones start to overlap and don't have the same sort of cytoarchitectural separation that they did in 6 zones or fewer. And so now we can kind of do this systematically across all numbers of zones, all people. And you can see that that you can kind of calculate the amount of overlap in individual zonation. And we find this very stark inflection point where right at the number 6 in the matrix cells, there's this huge jump, suggesting that 6 is a really nice number that represents the kind of molecular and spatial distinctions of these medium spiny neuron matrix cells. So the intriguing thing to us is that this zonation doesn't exist just in D1 matrix cells. It also exists in the D2 matrix cells. It also exists in the striosome population, which are this other population of medium spiny neurons that I mentioned earlier. And also, intriguingly, in certain glial populations, most especially astrocytes. That perhaps most remarkably, they show a very similar zonation pattern. Although they can't be divided into 6 groups, but only 4, those 4 really have the same kind of architecture and divisions that you see in the slightly more granular MSN population. So it really suggests that these divisions are consistent and coordinated across multiple cell types. So question is, what genes and pathways distinguish these zonal territories? And just to give you a brief introduction to that, Andrew Kraft has been looking at investigating the genes and signaling pathways that drive these striatal zones. And in the matrix neurons, the dorsal-most zone shows an enrichment of genes related to synaptic plasticity and dendritic growth, while in the more ventral accumbens adjacent zone, genes related to protein folding are relatively enriched. So meanwhile, in the astrocytes, a very interesting dichotomy exists where the astrocytes in the dorsal zone show a strong enrichment of TGF-beta signaling, which is known to be permissive for synaptic plasticity and remodeling, while smoothen and sonic hedgehog signaling dominates the astrocytes that are in the more ventral zones. So finally, just to kind of connect this work to the work you'll hear about from Steve, we were curious whether we could now impute the positions of neurons that had been sampled by ordinary, we call population single nucleus RNA-seq, utilizing spatial data as a kind of reference. And so here we're using the Allen Institute's MapMyCells algorithm to assign a zonal pattern based on gene expression signatures to these D1 matrix cells. And you can see that the population data for each zone really nicely matches the markers that we see. And so now we're in a position to ask, what's the impact of things like age on MSN populations? And when we look at this, we find intriguingly that the more dorsal zones show a stronger impact, transcriptional impact associated with age than the ventral zones. So this result just highlights the synergy of the two kinds of data our center are generating and how we can establish a kind of consistent, robust set of psychoarchitectural features in these cells and use them as anchors for understanding variation in our single nucleus RNA-seq data. Very briefly, just to say we're making these data into resources that the community can access. You can start to plot expression of individual genes across all of the individual spatial transcriptomics data that we've generated. And we're making kind of a viewer based on our whole mouse atlas framework to be able to do this. So I'll pass it off to Steve to tell you about the population work that our team is doing.
So unlike comparisons across species or differences from one cell type to another or differences across species, variation among individuals of the same species tends to be quantitative rather than qualitative. And that includes quantitative variation in the abundances of cell types as well as in the expression levels of each gene in each cell type. And so, and if you advance, Evan, so to measure this well and to try to get really just crisp, high precision measurements of everything, our team generates all the data using an approach that we developed actually initially as part of the NIH Convergent Neuroscience Initiative that we call cell villages, or in this case, we adapted it to nuclei villages, in which all the brain tissues are prepared together and all the nuclei from all the donors flow together through all the laboratory steps. And then using the software dropulation that was written by Jem Nemish and our team, at the end of the experiment, we re-identify every nucleus, reassociate it with the donor that it came from. And that's just what allows the kind of crisp, high precision measurements that you'll see enabling the analyses on the next slides. So we've used this approach to generate data on almost 4 million nuclei from 150 or so donors, actually different numbers in different brain areas. And if you go to the next slide, we just want to share one vignette from this work. Obviously, data like this makes it possible to look at many things, effects of aging and other characteristics. But today we're just going to focus on one thing, which is that the extent to which, for most genes vary in quantitative expression levels from person to person, and genetic variation near the gene often, at least partially, explains this in what are called cis expression QTLs. So this is, and this data was generated by our whole team, but in particular, all the analyses that you'll see in the coming slides were actually done by Tracy Yuan, an undergraduate data scientist on our team, mentored by these other team members here. On the next slide, this is one example of a cis expression QTL, and there are thousands like this, in which a common variant or common haplotype affects expression of a gene in essentially every cell type in the data set, including both all the striatal cell types as well as all the cortical ones. And mostly I just want you to see that when you collect data on this many people, and when it's when the measurements are precise enough, that some of these relationships, although these are not huge effects on gene expression, it's about maybe a 10 to 15% effect per allele, that it becomes possible to detect these things with overwhelming levels of significance when you generate data on enough people. And in fact, it's the number of people that's the main thing that determines power. That variant affects gene expression in all cell types, but we find just as many that affect expression in specific cell types. For example, this variant that selectively affects expression of the NPAS3 gene in microglia, and not in any of the other cell types that express this gene, even though many of those other cell types actually express the gene more strongly. This effect arises specifically in microglia. And I think when you think about it, it should be clear that you would never have known that this common variant that its effect arose in microglia just based on what cell types express the gene the most. You would have then thought it was like one of these neuron types. And then the same thing for this different expression quantitative effect, which arises in all types of MSNs, but in no other types of neurons or glia in the data set. And again, you would never have known just based on what cell type expresses it the most. You really need to generate data on lots of people to recognize what cell type the quantitative variation is acting in. Next slide. So we find such relationships for about 11,000 genes so far. You might wonder which of these kinds of patterns of cell type specificity arise over and over again. So here in the next slide, what we've done is for each of these 11,000 or so genes, and I realize that it looks like a lot less than that, but it's just because we're limited by the number of pixels on a screen. For each of these 11,000 or so genes, what is its kind of profile of cell type specific effects? And what you can see, and Evan, maybe you can just kind of step forward step by step, is that, for example, astrocytes have essentially the same distribution of expression quantitative effects, regardless of whether they're sampled in the striatum or the cortex. And if you advance some more, you can see that the same thing's true for oligodendrocytes and OPCs and microglia. That is to say that these effects tend to be the same regardless of brain area. But it's very different when we start to talk about neurons. So for example, there are thousands of MSN expression effects of common variants that aren't anticipated by any effect in any cortical cell type. And this points to the real value of looking in as many brain areas as possible, because each of these brain areas will have its own sets of neuronal types. So finally, I just want to present a couple of slides about some of the ways in which one could use a data resource like this to enable something that's been deeply needed in human genetics of complex disease, which is the ability to go deeper in trying to understand common genetic effects on those diseases. So as you probably know, these kinds of studies have been incredibly successful at mapping effects. There are now hundreds of common variants that are known to associate with schizophrenia or bipolar disorder, other brain disorders. But there's been this extremely common lament that it's been really hard for these to go beyond being vague associations because they involve non-coding regions and they have unknown effects on biology. That is to say, we don't know what genes they're regulating and we don't know in what cell types they're regulating them. So now with a data resource like this, and we'll hopefully have an online version of this soon for any common genetic effect, one can now ask how it manifests. And by how it manifests, I mean, in what cell types does it manifest? What genes does it affect? And what's the sign and magnitude of that effect? So Evan, if you just did, yeah, one more. Yeah, so, and then you can also ask these questions, not just for individual genetic effects, but collectively about all the genetic effects in a disease of interest. And we'll just conclude with an example for Alzheimer's disease as a handoff to the next speaker after us, Phil DeJager. So genome-wide association studies for Alzheimer's disease have mapped more than 70 risk variants, almost all of the non-coding regions of genes. And we can just ask how those effects distribute across these sort of 13 clusters of cell type specific effects from these 11,000 or so such effects that we've found so far. And what you can see is that the effects in Alzheimer's disease concentrate in this cluster, cluster 13, which involves effects in microglia and potentially other monocyte populations, kind of aligning with other things that are being discovered by other kinds of approaches in this disease. I just want to thank the whole team, the whole interdisciplinary team that generated all these data and did these analyses and is working hard to now expand it far beyond the cortex and striatum to the rest of the brain. And I just want to conclude with one other slide, actually. This is about, is there one more slide? No, okay, sorry. No, you can end, Evan. That's, we're done.
Great. Thanks very much, Steve and Evan. We have time for one quick question. If you'd like, please use the raise hand icon on the top of your screen.
Evan, it's a question in the chat. You showed the coronal, these nice clusters. What happens in the sagittal? Does it retain sort of a cylindrically symmetric thing, or does it, have a different kind of pattern?
That's a great question. I think we started because we wanted to have we started with this set of slabs on the anterior side because we wanted to be very consistent. But now the team has been doing some more posterior profiling, so we should be able to know whether how the evolution of these profiles, these zones evolve over the sagittal plane. Both the primate and the human data samples that we have are all coronally sectioned, so we do need to do it from the slabs. But one thing I didn't mention that is kind of encouraging to us is that we see the same zonation when we profile the macaque as well. So we think this is conserved in non-human primates as well.
And mice comparison?
We don't see it in mice.
There are some zones in the striatum in mice.
There are, but they don't seem to follow the same pattern of the ones that we see.
Okay, so that's very interesting because the striatum is supposed to be so conserved, and now you're saying it's actually different.
Yeah, more to come on that.
Great. Thanks very much, Steve and Evan. In the interest of time, we'll have to move on, but please, people, feel free to carry on the conversation. There's a chat icon right at the top of your screen, and you can continue the dialogue with Steve and Evan that way. Thanks again.